All right, so we continue on with our discussion of protein function, and where we're going to continue is by talking about a different form of hemoglobin, and that is fetal hemoglobin. Fetus receives oxygen from its mother's blood via exchange through the placenta. Now, fetal blood has a higher affinity for O2 than the mother's blood. Now, why is that? And we can look at this oxygen binding curve over to the right here, which shows basically the binding curve of fetal hemoglobin versus maternal maternal hemoglobin. And one of the things that I want you to recognize about this is that our curve from maternal to fetal is shifted to the left. Now, why is that? Well, fetal hemoglobin has a different composition. Rather than being 2 alpha subunits and 2 beta subunits, making a tetramer, which is the case with wild type adult hemoglobin, HbA, HbF has a composition of 2 alpha subunits and 2 gamma subunits. Now, ultimately, so the intrinsic oxygen affinity for fetal hemoglobin is very similar to that of adult hemoglobin. Fetal hemoglobin has a much lower affinity for bisphosphoglycerate than adult hemoglobin. So it has a lower affinity for O2, in which case there's a stark difference between these proteins, and what's the rationale or what's the reason for that? Well, adult hemoglobin, HbA, has a histidine at position number 143. Now, one thing that's important to remember about histidine, and this goes back to amino acid structures, histidine is a polar positive amino acid. And this is in position number 143 at the beta chain. Well, fetal hemoglobin has a serine at that same position in its gamma chain. So basically the same position, but a slightly different protein. We have a serine, which, keeping in mind what serine is, S-E-R, is a polar uncharged residue. So it's important to remember the significance of these changes. So polar positive to polar uncharged residue, sure, they're both polar, but think about the types of interactions. Histidine is able to stabilize an ionic interaction or definitely participate in hydrogen bonding, whereas a serine residue, that's basically only able to establish a hydrogen bond. Now, thinking about the strength of those interactions, well, hydrogen bonds are weaker than ionic bonds. And so that's one thing. In addition to that, just the types of interactions and the amino acids that are going to come in contact with this other, or with like a positively charged histidine as opposed to a polar residue. The charge of a positively charged histidine for a neutral serine ultimately is what is going to cause a reduction of the binding affinity for bisphosphoglycerate. Because if you remember bisphosphoglycerate, it had a total of 5 negative charges. So those 5 negative charges, in the case of bisphosphoglycerate for histidine, well, it's losing out on those interactions. Now, it's not uncommon for adult hemoglobin to have some mutations. And in many cases, those mutations don't really have any significant implications. However, there are variants that ultimately alter hemoglobin's quaternary or tertiary structure. In this table on the right, there's a series of different hemoglobin variants that are implicated or identified. And what's described here with this mutation is what amino acid in which subunit, where that amino acid is, and what it's mutated to. So let's just go ahead and take a look at 2 of them. Let's look at Hammersmith and Milwaukee. And let me go through what the rest of these mutations do. They can destabilize hemoglobin to the extent that red blood cells lyse, causing what's known as hemolytic anemia. They could favor the oxidation of Fe2 plus as opposed to Fe3 plus, which causes cyanosis due to the increased concentration of methemyoglobin. Now, this methemyoglobin is basically you're dealing with a low oxygen containing red blood cell. But let's go ahead and look at these two mutations, Hammersmith, which I'm going to label H. Smith, and Milwaukee. Okay, so Hammersmith, and yes, these are named after cities. I believe that's the case with all of these hemoglobin mutations are named after locations in which they're found, where they may be first discovered. So we are dealing with a Hammersmith mutation on the beta subunit, a phenylalanine F42 is mutated to S. So that notation that I'm using there, F42S, indicates that phenylalanine in the wild type, or in its normal, so-called normal hemoglobin, is a phenylalanine at position number 42. In this mutant, it's mutated to a serine. Now, Milwaukee, we're again dealing with the beta subunit. Valine 67 is mutated to GLU, or glutamic acid, which is going to be E. Now, what I want to do is really just show let's talk about those specific mutations and what those amino acid differences are. A phenylalanine, well, that's an aromatic, polar, non-polar amino acid, non-polar aromatic, going to a polar amino acid. So there's a couple of things to think about. One, we're switching basically the polarity of this amino acid, the types of interactions that it's going to participate in. Something that was going to prefer a hydrophobic interaction now all of a sudden likes hydrogen bonding. In addition to that, think about the size of that backbone or the size of that R group. With a phenylalanine, you have something that looks like this, CH2. Then we've got a ring. Looks like that. Versus a serine, which looks like this. And those aren't really drawn very well to scale. The molecular weight of a phenylalanine R group versus the molecular weight of a serine, they're going to occupy different spaces, in which case the types of interactions that they will participate in is going to be different. Now, Milwaukee, we have a valine mutated to a glutamic acid. Well, valine is a non-polar, it's a non-polar amino acid. That's just a hydrocarbon side chain. Glutamic acid, on the other hand, is a polar negatively charged amino acid. So again, sort of switching the polarity of the amino acid in that particular location. Now, what the kind of downstream effects are is this Hammersmith mutation that is going to weaken heme binding. The Milwaukee mutation is going to promote methemyoglobin formation. So we have two things that are ultimately going to diminish hemoglobin's ability to bind oxygen, but through slightly different ways. And this is an instance where, man, if you profile all of the different mutations, they might all have something very similar to, well, they diminish hemoglobin's ability to bind oxygen, but that's only like one layer of the story. You've got to get a little bit deeper and say, okay, why? What specifically is happening? Is it something that is, it's causing a different oxidation form of iron to bind to hemoglobin, in which case that's causing a reduction in O2 binding? Why is that? And it's whatever you get deeper into that, then you have a better response and a better understanding of, well, how is this treated? Is there a drug or is there some sort of therapeutic intervention that can be used for this? But I wanted to show you what this specific notation was, which is the normal or wild type amino acid, and that would be the first one that's listed. The number indicates the position, and that last letter is indicating the new amino acid in its place. So since hemoglobin is kind of a involved protein, this mutation notation with CD1 42, that's just showing more information about where that amino acid is with respect to all of the different alpha helices. Because if you remember, hemoglobin has like 8 different alpha helices, and they are labeled A, B, C, D, E, and I think F might be the last one. At any rate, I'm going to proceed from here. So one mutation that you might be fairly well familiar with that we already kind of alluded to and that we mentioned when we were talking about the significance of primary structure of a protein is sickle cell anemia. So sickle cell anemia is something that people are maybe a little bit more familiar with the trait, and they're maybe a little bit more familiar with what the grand implications are. Excuse me. And they may even know someone who has that as something that they live with. Sickle cell anemia has been tracked back to a single valine mutated to glutamic acid in the beta subunit. I cannot remember the number. We looked at it in a previous presentation. And deoxy hemoglobins, that valine binds in a hydrophobic pocket of another hemoglobin. So insoluble filaments that deform this HBS is indicative of our sickled hemoglobin. So what we have is insoluble filaments that are forming. Now, the oxygenated sickle cell hemoglobin, the valine is not exposed, so those filaments do not form. So the deoxyhemoglobin S filaments dissolve upon oxygenation, so no fibers are present in that arterial blood. But ultimately, what we have is these uniquely shaped proteins or uniquely shaped red blood cells that. So these sickled cells, they form these complexes that they otherwise should not form. Or these sickled, red blood cells ultimately aren't able to carry as much oxygen, whereas the hemoglobin mutants lead to these fiber formations, which lead to ultimately these sickled cells. Now, sickle cell anemia and fetal hemoglobin, well, some sickled hemoglobin homozygotes, so when somebody has or both of their genetic copies of hemoglobin have a sickle mutant, they might show only a mild form of anemia. And that's because they express high concentrations of fetal hemoglobin. Now, hydroxyurea administration as like a drug induces the production of fetal hemoglobin. So that's one intervention that can be taken. Now, interestingly, if somebody is afflicted with sickle cell anemia, they are much more resistant to contracting malaria. Now, malaria is caused by a mosquito-borne protozoan, which resides in the erythrocytes during its life cycle or its 48-hour life cycle. Infected erythrocytes adhere to capillary walls, causing death by impeding blood flow to organs. Now, the protozoan causes a decrease of pH by 0.4 units. That's no small change. That's a very significant difference. The lower pH favors that deoxyhemoglobin. The parasite ultimately induces sickling. The parasite-induced sickling allows the spleen to remove the infected erythrocytes from the system. And that's where I'm going to go ahead and stop with with that material, we're going to jump into the next in just a moment with muscle protein.